Today I have another more application-oriented talk, and I'm going to talk about challenges for graph theory in human neuroscience. So I'm Katelyn Arnemann. I'm a PhD student in neuroscience here at Berkeley, and I work with Dr. William Jagus on human aging and Alzheimer's disease using neuroimaging methods. And today I'm going to broadly introduce some of the challenges for graph theory in human neuroscience. The goal of my talk is to get you all thinking about the sorts of questions that graph theory can help answer in neuroscience. That is, how graph theory can be used or has been used in the field so far, as well as challenges we face in defining and interpreting graphs. So conceptualizing the brain as a network has revolutionized the way that we think about and study brain function. Graph theory has allowed us to characterize and quantify how different brain areas influence one another in health and disease. And so this is kind of in like an alternate framework that allows us to really quantify and look at these interconnections. That is basically how the brain truly functions. Graph theory has allowed us to characterize the topology of brain structure and function, which has been important for helping different concepts emerge, such as the existence of highly connected hubs that may serve an important role in coordinating brain function, as well as communities of brain areas that work together and form modules, and maybe help achieve specific cognitive functions like vision or speech. We have also been able to examine similarities and differences in how distributed brain areas coordinate to achieve different cognitive functions, with graph theory enabling us to begin to understand how differences in the structure of coordinated brain function underlie the computations being performed. While we're still in the early days, this has given us a tool set to begin to understand human cognition. And so here we could just see like adjacency matrices for a bunch of different tasks, and we're starting to understand and examine how are these similar, like so maybe there's different computations the brain is performing that's similar across tasks, as well as different, and we're beginning to examine this, but it's very challenging to gain meaning from this approach. Finally, it's being used to understand and characterize different neurological diseases. For example, the topology of a brain graph, such as this, can be used to generate hypotheses, for example, about differences based on nodal stress, based on total flow through the network structure, or how it might spread from one region to another through a network structure, and differences in things like trophic failure. So, how might trophic failure influence, a more central node versus a node on the periphery. And we can use these to make predictions about which brain areas experience degeneration or atrophy, like due to neural shrinkage or death in something like dementia. And so this is more along the lines of the area that I'm currently working, although I've also worked in more cognitive applications. And so you'll hear a little bit more about how I'm using models a little bit like this to examine aging and dementia. So here are just kind of a few really broad categories of challenges for neuroscience. So first I'm going to go through kind of the most basic aspect, which is just defining the elements of a brain graph, which is where we all start, but actually has a lot of challenges. And I do think a lot of people get stuck on this part, because there's no, in many cases, especially given the methods in human neuroscience, there's no like ground truth that we can look on, and we just have to like accept the arbitrary nature of some of our decisions. And then I'll move on to something that I think is more likely to benefit the fields, which is how do we compare graphs and the tools that we can use to do that, as well as some graph properties and algorithms, where I'm mostly going to focus on applications in my own research, just because there's so many different choices. And so there you'll get to see one of the problems that I'm working on right now. So as I've mentioned, healthy brain function requires coordination across distributed areas of the brain. And so what might come to mind are two neurons that are directly connected and coordinate electrical impulses through a synapse. And maybe we can establish their connection directly by imaging the neurons and seeing that their axons, one axon reaches to another, or indirectly by stimulating one neuron and measuring a response in another and getting some causality. And so this is like the ideal case that basically we can never do in humans. And so this is where like a lot of the challenges come in. And even in this like simple case, it gets complex because the neuron only responds with some probability and so on. And so a lot of this isn't as straightforward as we'd hope. And so what happens when we get into humans and we can't use these invasive measures that maybe are more easily interpretable and have more easily tied to some ground truth? All right. Oh, so we're going to focus here on human neuroscience. All right. So the first challenge that I'm going to talk about are what are the elements of a brain graph in terms of the nodes for humans. So in many cases, the methods of data collection determine the nodes. For example, it may be voxels, which are 3D pixels, which is what we get from MRI and PET neuroimaging, or it may be electrodes in the case of something like EEG, which could either be on your scalp or directly on the brain in very special patients that undergo surgery. And however, sometimes there's tens or hundreds of thousands of, say, voxels from neuroimaging. Sometimes we decide to group them into what we call like regions of interest, or I'm just saying brain areas. And one of the issues with this is that there's tons of different ways of grouping them. You could group them based on having similar anatomy, but there's many different features of anatomy. It could be cell type, it could be genetic profile, it could be just location or spatial proximity, or based on their function, like maybe they operate in a coordinated way or respond similarly to some stimulus or whatnot, what have you. And so this leads to many different challenges. So as I mentioned, there's many different ways of defining the ROIs. And I think that this is where, like in my experience of collaborating with and trying to start collaborations, a lot of people put their attention in. Like, I feel like computer scientists really like this problem, and it's hard to move past. And which I think is clearly very important, but also, right, there's like about 1 million different atlases proposed, and sometimes if you want to understand what the brain's doing, you need to just choose one and see what you get with it, or try maybe multiple atlases and so on. And so there's no ground truth for this. And so that's one aspect. But also, we don't always know what, or we can't measure what the correct or ideal level of measurement is for, examining something like as abstract as cognition or even motion or many different things. And so we often have to, either stick with what we have or make some guess about, maybe do synapses matter? Like, do we have to study it at that small of a level, or can we, zoom out and gain insights? And I think a lot of these different levels have different insights to be made about how these operations are performed in the brain. Yeah. So each neuron has like tens of thousands of synapses. I mean, it varies a lot by the neuron, but there can be thousands of synapses on one neuron, and one neuron might synapse onto multiple neurons, and it really varies by their type and so on. And I mean, in humans, we can pretty much never get at that level, and sometimes when you're even in animals recording, you don't know the type of neuron that you're recording from and so on. And so you have to kind of make guesses. Sometimes if it doesn't seem like it's, you might get an inhibitory neuron or something, and people just are like, oh no, we don't want to record from that. And so there's a lot of choices. So yeah, synapses are very small. It's small, just a small part of a cell, versus ROIs might be like a huge, a fist-sized part of your brain or something. It depends on the measure. And one other problem that, depending on the method you might face, is that there could be like inconsistent or variable structure across individuals or groups, right? People have brains that are different shapes, so if you're looking at it, even at the smallest level, you have to somehow combine this information if you're recording for multiple individuals. And also, you can sometimes just not have complete brain coverage for certain measuring methods, and they may not be like positioned in exactly the same spot, depending on the method. Right, and so the second part, defining an edge, this is kind of more maybe obvious to a lot of you, but there's many different ways you can define it. It could even be a structural connection, or it might be some functional time series correlation between pairs of nodes, or partial correlation, or lasso. Most typically, I would say correlation, just a simple Pearson's correlation is used, and I think that's, like, partial correlation can get problematic when you have not that many time points and lots of different nodes, and so it's a continual challenge. But so typically you think of it over time, which is what we would get from like resting state or functional MRI, but sometimes, and I'll be talking about that a little, I guess I'm not for this, but sometimes we do it across individuals. So we have different individuals, and we're looking at like how, the thickness of certain areas of the cortex co-vary across people. And we look at that, say, development, with some idea that areas, develop at the same time and have similar thicknesses, or they degenerate together. And if they're degenerating, at similar rates, maybe they're connected. And so there's just different assumptions you can make with that. So challenges, sometimes the measurements are interdependent. Not even, like, it may not even be because they're communicating, but it could be because, the person's moving during, and that causes artificial correlations. Or, for PET, it's unclear, like the resolution and the signal isn't high, and so there can be correlations with, between the voxels at that level. Like getting a directed edge is really challenging, given that there are loops and we often can't stimulate. And there's also issues with deciding on a threshold. So thresholding the graphs if you're going to have it weighted or binary, and so on. And so all of these I really mostly introduced just to kind of preempt the next challenge, which is how to compare the graphs. And so, right, people define graphs, researchers define graphs in all these different ways. They use different analysis. They use different, some use partial correlation, some use correlation. There's different modalities. And so there's all these different factors going in, and then they're trying to say some general insight about brain function, but now there's all of these different sized graphs and constructed in many different ways. And so that makes it very hard to compare them. And then there's also kind of inherent, non-researcher created problems, like, maybe different people or populations have different ideal numbers of nodes, or, ideally you would threshold it at a different level or something. And so a lot of graph metrics are challenging to compare in graphs, say, with different distributions of weights or different numbers of nodes and other differences that make it hard to compare them. So, comparing metrics across diverse populations and different modalities is a continual challenge. And so, just to say, there's a lot of different graph properties and algorithms that aren't either have like inherent issues like community detection, unless there's truly completely separated communities. Often there's not like one clear answer. And these differences in the results of, say, a community detection algorithm can lead to issues in one comparing studies and interpreting them, or also just as a result of maybe people are creating these algorithms in this case where there's obviously separate communities, but in neuroscience, we maybe think that some parts of your brain are parts of multiple networks or multiple communities, and so how do you allow for overlapping communities? And so there's many different kind of challenges like that, and I'm going to talk about one today that I'm currently contemplating. And so this is definitely a work in progress that would be fun if anyone has ideas of approaches I could try, it'd be fun to hear those. But just to briefly introduce what we're looking at here. So Alzheimer's disease is typically confirmed at autopsy based on the presence of two different abnormal proteins. One are these amyloid beta plaques, and the other are the neurofibrillary tangles called tau. Probably not going to go too much more into it, but so the data I'm looking at here is from PET, and here we have a radio tracer called Pittsburgh compound B that we inject, and it binds to the similar sites. It binds to the amyloid beta plaques, and through detecting its emissions from the radiation, we're able to locate with some not great amount of precision the distribution of this signal in someone's brain. So this is an example of an image we would get from an Alzheimer's patient where the hot areas are areas with high signal. And so this is all the areas you can see is very widespread that are affected by amyloid plaques. And here's an example from just a normal older adult. But we also see and are really interested in these healthy, cognitively normal older adults who have almost comparable or high levels of amyloid pathology that can be similar to that of an Alzheimer's patient. And so say if you look at people around 70, about 30% of them have this signal. And we're not 100% sure what that means. Like, would they all eventually get Alzheimer's? I mean, I think so, but you can't really prove it. But so there's some really over the course of decades long trajectory of developing this disease. And so it's really hard to say, at what point this person's going to transition and become cognitively abnormal and face memory issues. And so one of the really neat graph-related insights, I think, is kind of studies after studies showing that the neurodegeneration and many different types of dementia, so age-related diseases of the brain, seem to follow like a network-like structure that's kind of unique to each brain and emanating from different nodes on, say, a graph. And so here, just to look at a Alzheimer's patient, here are the loss of cortex, so cortical thinning, in blue. And then if we look at from the area with the most thinning, so the most degeneration, in younger adults in different ways, two different ways here, and you see how it's either across structure, so using the structural covariance method, or in terms of the functional synchronization from the seed, you could see that it correlates with the areas that it synchronizes with are those that later get the disease, which I'm explaining that the best way. But so there's some evidence that areas that are synchronized degenerate together in these diseases. So maybe they're spreading through some network. So here is kind of progression of the two primary pathologies in Alzheimer's. Here's tau, and here's amyloid. And here is just kind of a concrete thing, like there's different stages of where in the brain, say, tau accumulates. And this also seems to be true, although much more complex for amyloid. And so this is like a GIF with going across individuals with increasingly high amounts of amyloid, and it seems to build up in this characteristic spatiotemporal pattern. And that's what, in this project, I'm interested in modeling using graph approaches. And together, these patterns, spatiotemporal patterns of accumulation of them suggest that there's differences across the brain in vulnerability to amyloid and separately to tau. So I'm going to go through two different ways I'm looking at this, because I'm still kind of trying to figure out the best way to get at this question. It seems to me, or we seem to think that amyloid, unlike tau, that there's some evidence that it starts in like a very specific brain region, it spreads through neurons, though this is still being tested, but to connected areas, it's more like up in the air for amyloid, and maybe it seems to start and spread from multiple epicenters. And that's one way to kind of just simplify the problem as much as possible is I started by looking at a minimum spanning tree. So I'm looking, in this case, it would reflect the path of maximal information flow through the brain. So I have some graph of brain areas and how synchronized they are. And then I'm just trying to reduce it to the highest connections. And I'm not going to go through this since you may or may not be familiar with this, but you get some tree extracted. And in this case, it reflects the brain, the edges that are maximally connecting all the nodes in the graph with the minimal number of edges. Right. And so here is just like plotting this graph that I got in the young adults. And what it looks like, so I'm going to try to look at how amyloid spreads along this tree. And then here's it kind of in the side of a brain space. I should have put a picture. But so it's like, this is the front of the brain, the back, the bottom, the top. And so just to kind of see what that looks like in this particular case. And so the way that I'm going to look at the pathology is I'm looking at this across people, where I'm looking at those with increasing amounts of total amyloid pathology. And so here are those with very minimal pathology. And here's the distribution as I go across to those with really high load of pathology across a lot of the brain. And so I'm going to be trying to kind of model this on a graph. And so here's where now each of these is a brain region. I'm just going to put each of these as a node on this graph and extract the amyloid value. And so that's what you're seeing here. Not sure how visible it is, but so I kind of see that, you know, like there's like this cluster of high amyloid areas right here, and then that continues there, and then maybe something kind of pops up here, and then it's kind of spreading up here, and so on. And so to me, this felt like, or looks like, right, maybe there's like multiple points on this graph, and then it's accumulating in the neighbors through some way. And so the challenge that I've been trying to figure out is how to, you know, quantitatively decide what such an epicenter might be or define. And here I just kind of did like some heuristic approach where I'm like, okay, you know, it's higher in an epicenter, and then, you know, its neighbors get more amyloid as the time goes on, and they have to be some distance apart or something. It's kind of a hacky approach, which is why this is the final project of my PhD. It's been the hardest for me to come up with approaches that I'm happy with. And so here's just kind of a GIF of it, but in the interest of time, I'll let it go through once, but just to show what this particular network looks like. And I've switched now to a larger model with more nodes, which has made it more complex. And just to really quickly go through a different approach that I'm not sure if I'm going to use the tree or not, but I started thinking about like, okay, maybe it would be easier if I had directed edges in some way, and then I could just define an epicenter as like, you know, nodes with high out degree and no in degree or something. And so here I'm looking at, say, node A at some initial time point where, all right, over time, this node has higher pathology with some slope that I could define. And then here we have this for now a bunch of different nodes at a couple different stages. And I can look at, and try to look at the slopes of these and create some notion of directedness, which I believe his name is Eric Friedman at Berkeley here, is an affiliate here, came up with this method. And so here we can see, all right, this node has a high slope. And so in this approach, we require that a node is increasing, so it's accumulating pathology over time at this stage. And then we're looking for in the next stage which nodes now accumulate more than they were, so they have a higher slope than they did before. And so we would call those like accumulators and draw maybe something where like this node's increasing, now this one's increasing more, and maybe there's some causality going on. And then we look at a next stage where, all right, now this node went from negative to positive, and maybe since this one just started accumulating more, maybe there's some directed edge here. And so that's kind of the general gist of this approach, but it requires, I'm trying to figure out one of the things that I've been thinking about is what these slopes should look like, and it's actually influenced by the way the PET tracer is taken up in the brain, and it may be tracer dependent, and there's actually, it's nonlinear, as you get a ton of it, there's less brain for it to, maybe your brain's atrophying and there's less brain for it to attach to, and so the slope can go down actually once it has really high amounts of tracer. And so I've been kind of trying to think about the best ways to, to model what we would expect something to be sudden, starting to really accumulate versus something random. And so this is here's an example of the network X directed edges thing where I still need to think about, maybe it's getting a little dense and hard to see here, but here I've plotted the directed edges that I got from this approach. And here's where people have very low levels, but I'm going from people with really low levels to those with maybe borderline low. And then here's, transitioning from people with substantial amyloid to getting even more. So how are these stages, causing increases? And so I'm trying to plot that out here, and the nodes are just colored by their out degree, where I removed I set to 0 any node's out degree if it had already in degree greater than 1. So I'm looking just for nodes where it seems to be spreading without much input, but that is still a work in progress. And so finally, just to say here I just kind of hand chose these stages of progression, but maybe there's some data-driven approach. So here's the total load in the brain versus the load of pathology in one region, and here's some slope of it increases, and this is actual data and what it looks like. So maybe somehow looking at the data, we could choose the point at which it really seems to start accumulating. And so that's a different idea I've thought of. And so this is maybe my multi-pronged approach of how I've been struggling to best model this data. And with that, I just want to thank my lab and you all. And I look forward to talking with you after. Thanks.